Chapter 9 is muscle tissue. We will go through the microscopic anatomy and the molecular anatomy of muscle tissue, and we will look at the physiology of muscle tissue in terms of how do muscles contract. We will not be covering the gross anatomy of muscle. That would be Chapter 10, and that would be done in anatomy and physiology too. So let's just review the main types of muscle that we see. We looked at these in Chapter 4 in the tissues chapter. In the tissues chapter, we saw that we have skeletal muscle, and that skeletal muscle shown here in a photomicrograph. The skeletal muscle is also called striated muscle, and that has to do with these stripes or striations that we see. Today, we will spend a lot of time talking about the molecular organization or anatomy of these muscle cells so that you can understand what those stripes are. Okay. Those striations are going to be critical in terms of how does this muscle actually contract. Then we have cardiac muscle. Cardiac muscle is also striated. You see some striations. In addition, you see these much darker areas that are called intercalated discs. This cardiac muscle is only found in the heart. It is very unique. There are a lot of very special qualities that cardiac muscle has. We will be focusing mostly on skeletal muscle, but we will talk briefly about cardiac muscle. And then finally we have smooth muscle. Smooth muscle is going to be the type of muscle that will be involuntary, and we'll see in our smooth muscle in the hollow organs of our digestive system and our reproductive systems and things like that. Now there are similarities between all of these muscles. Okay. All of the cells can be referred to as muscle fibers. Okay, the cells are elongated, so when we talk about muscle fibers, we can use that interchangeably with the term muscle cells. Muscle contraction is going to depend on two kinds of myofilaments. Whenever you see muscle terminology, there are certain key parts, key prefixes, okay, and those prefixes that are important are myo, mice, or sarco. So if you see myo in a word, it means muscle. So a myofilament, this is going to be the filament of the muscle. The muscle contraction is going to be dependent on these myofilaments, actin and myosin, and these are proteins, and we will look in depth at the structure of these proteins and how they interact. Some of the specific terminology that you need to get right off the bat. Here we use the term sarcolemma to describe the plasma membrane of a muscle cell. The plasma membrane of a muscle cell is very special. Okay? It's very unique in the way that it behaves, and that's what allows muscles to do what they do. We also have the sarcoplasm, and the sarcoplasm would be the cytoplasm of the muscle cell. So we have those prefixes of sarco or myo, and that's going to let us know that we're talking about something with muscles. So if you see myo, you see sarco, immediately think muscle. Now the skeletal muscle tissue, if we look at a single cell or a single fiber, you should note a few things that are unique about it. Okay? Those striations are present. There's also multiple nuclei. This is one of the few times that we will talk about a cell that has multiple nuclei. This skeletal muscle fiber would be found within a much larger organ, and we will talk about the organization of the skeletal muscle into organs. And this is going to be under voluntary control. This is going to be attached for the most part to the bones, and it's going to allow for locomotion. Now these have very rapid contraction, but they tire very easily. And this is responsible for the overall movement and motility of the body. The cardiac muscle is only going to be found in the heart. We do see those striations. Cardiac muscle is not under voluntary control. In fact, cardiac muscle, its inherent beating or contraction is not even under the control of the nervous system. When you talk about the cardiovascular system in A&P2, you will learn that the heart has these very unique cells called pacemaker cells. And those pacemaker cells can induce cardiac muscle cells to contract without any interaction from the nervous system. Now, the nervous system can speed up or slow down those inherent contractions, right? If you get scared, your heart races, right? If you meditate, your heart slows down. Those are ways that your nervous system can modulate the contraction of cardiac muscle, but it can't start it or stop it. Then the smooth muscle. Smooth muscle is going to be found all along our hollow organs, our viscera, so the stomach, the bladder, the respiratory passages, the arteries. This is not going to see, you will not see striations in this. It does contract. When we look at the organization of smooth muscle tissue, we will see in those hollow organs, we will have 2 layers of smooth muscle tissue, and that dual layer is going to allow smooth muscle to function. Overall, smooth muscle tissue contracts slower, but it doesn't tire as easily. So your stomach, your intestines usually don't get tired of digesting food, right? Whereas if you walk for a long period of time, your skeletal muscle does get tired. It actually starts to tire out. So all of this muscle tissue, whether we're talking about smooth or cardiac or skeletal muscle, have certain things that are their functional characteristics, right? All muscle tissue is going to do certain things. One of these is the ability to be excited. The other word is to be irritable. Okay. I choose excitability. And what excitability is, this is the ability to receive and respond to stimuli. This is going to be a very important term, both in this chapter and also when we talk about the nervous system. Okay. When we talk about how muscles and neurons can become excited. Muscle then also has the ability to contract, and contractility is the ability to shorten the length of the muscle fiber forcibly. Right? And we will look at the mechanisms involved in skeletal muscle in terms of how does that contraction occur, right? How does contractility occur? The other thing that muscle has to be able to do is it has to have extensibility. It has to be able to extend beyond its resting state. Okay? That's stretching. And then the ability to move from a state of being contracted, relaxed, and stretched or extended, going back and forth, that's elasticity. Okay? That's the ability to recoil and resume resting length. So when a muscle is stretched, it can return to rest. When it is contracted, it can return to rest. So all of the muscle tissue that's functioning properly will have these different functional characteristics. I've mentioned the different functions. Try and keep these functions in mind in terms of what the different types of muscles do. Right? So skeletal muscles is what we tend to think of as muscles. They're responsible for moving our bones, things like locomotion. The cardiac muscle makes the heart beat. But why? What is the point of that? Right? That's to propel blood through the body. Okay? The heart is a pump. Without that pump, your blood doesn't travel through the body. Blood doesn't travel through the body. Your body cells die. Your smooth muscles can help to propel stuff through your body. So they propel blood through your body. They help to propel food through your body. They help to propel wastes out of your body. And then there are some side components of this, where muscles are also going to be able to maintain posture, stabilize joints, generate heat. Right? When we looked at the joints in Chapter 8, a lot of the joint function is due to the muscle tone, right? The tendons that actually cross those joints. Right? We looked at homeostasis way back in Chapter 1. We talked about body temperature regulation. Right? The muscles, the contraction of the muscles actually helps to regulate our body temperature. Okay? So there are some more distinct functions of muscle we have to think about. So let's take some time looking at the anatomy, specifically looking at the structure and the organization of skeletal muscle. The problems here is the word muscle can be used at many levels. Okay, so I will try and get you to start thinking about that organization that we have, the organization of life from way back in Chapter 1, and use those terms. Okay. So if we talk about the organ level, this would be our highest level, the biggest level. right? So if we were to talk about the biceps muscle or the quadriceps muscle, okay, that is an organ. Remember that the definition of organ is where you have different tissue types working together for a function, right? So here we're going to see there are different tissue types because we're going to have muscle tissue, we're going to have connective tissue, and the function is move a bone, right? So the biceps muscle is an organ. Now looking at this generalized organ, right, we can see that it's broken down into smaller and smaller parts. So we will look at the structure in a top-down way, right? So this entirety of this muscle is going to be surrounded by connective tissue called epimysium. Right? Within this is actually going to be these bundles. Right? Each bundle is called a fascicle. If we were to focus in on the fascicle, which is a portion of the muscle here, okay, we see that the fascicle then is a bundle of muscle cells or muscle fibers. This fascicle is going to be bounded by connective tissue. Here we have perimysium. So this bundle of the muscle fibers are the muscle cells. Here we're talking about the individual cells. So we'll focus in on an individual muscle fiber or cell. And we'll see that this cell, it's a skeletal muscle cell, so we'll expect to see multiple nuclei. It will have a layer of very, very delicate connective tissue surrounding it called endomysium. And then it's a cell, so it must have a plasma membrane. And the special term we use for plasma membranes of muscle cells are called sarcolemma. Now this muscle cell is very unique because it has within it these complex organelles that we don't see in really any other cell except for muscle cells. And those complex organelles are called myofibrils, right? Notice that there we go, myofibrils. These are going to be organelles that are specifically designed to allow for muscle contraction to occur. If we focus in on that myofibril, we'll see that it is composed of bundles of myofilaments. Now the myofilaments we previously defined as being the proteins that are involved in contraction. We said actin and myosin. Right? If we take a look at a myofibril, it's actually made of these repeating units that we call sarcomeres. Right? Now this sarcomere, if we look at this sarcomere, it's a segment of a myofibril. Very, very, very important. This myofibril segment, this sarcomere, this is where we're actually going to see contraction occurring. So when we talk about how contraction occurs, we're going to look at it at its smallest level here within a sarcomere. And we're going to look at the specific anatomy of the sarcomere a bit more in depth. And what this sarcomere is made of, it's made of those myofilaments. So if we were to focus in on just a little piece of the sarcomere here, we can now see those myofilaments. And those myofilaments are proteins. They are going to be called actin, right, which is shown here as this thin filament. In these diagrams, it's often shown in blue. It's not actually blue. Then we also have the thick filament, and this is myosin. And these two proteins interacting together are going to lead to a contraction. Okay? And that contraction here is going to make this sarcomere contract. And if this sarcomere contracts, we'll see that there's a very tight interaction between skeletal muscle and the nervous system. And that is going to allow all of the sarcomeres within a myofibril to contract. And it's also going to make all of the myofibrils contract at the same time. And all of the myofibrils within a muscle cell contracting at the same time make the muscle cell contract. And if all of the muscle cells contracting at the same time within a fascicle contract, then the fascicle contracts. And if all of the fascicles within the muscle organ contract, then the muscle contracts. So when you see the contraction of an organ like the biceps muscle, what's really happening is you have contraction occurring at the smallest level as an interaction between actin and myosin, making every sarcomere contract, which makes every myofilament contract, which makes every muscle fiber or muscle cell contract, which makes the entire fascicle contract, and all the fascicles contracting together makes the muscle contract. Right? So that's a lot of coordination. Right? That's a lot of coordination. That's like trying to do the wave across the world. Okay? And so what we're going to see is for this to occur, we're going to have a really, really good way of making all those contractions occur at the same time. A high degree of organization. Okay, so as I was going through that organization, I mentioned these connective tissues. So I want to just clearly state these. Okay. The outermost connective tissue is called epimysium. Right? Epi meaning outermost or on top of, mice having to do with muscle, and then eum. Remember that eum? Right? That means covering of. We had periosteum, right? Perichondrium. Okay. So now we have epimysium. Okay. This epimysium is going to be this outer covering. It is going to be dense regular connective tissue. It's actually continuous with the tendon. The tendon is the dense regular connective tissue that connects the muscle to the bone. Okay. Within this organization, we have these bundles. Each bundle is a fascicle. Okay. These fascicles are going to be bound with perimysium. Peri usually means in between. This is also a fibrous connective tissue that's going to surround the groups of muscles. And what you notice in this diagram is in addition to having fascicle, 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 there are little spaces, and that's where you'll see blood vessels and nerves passing through. The final layer is the endomysium. This is the innermost. This is a fine sheath of connective tissue, and this would be surrounding each muscle fiber, right? Each muscle cell is the endomysium. So you do want to make sure you keep those different definitions and terms straight. Now we're going to take a little bit of a closer look at the myofibrils. These myofibrils, what we have here, are going to be these densely packed rod-like contractile elements. This would be a myofibril here. So what we are seeing, this entire thing then is a section of muscle cell. This is where we see the level of the striation. So when you look at this under the microscope, you're seeing the cells. In those cells, you see striations. You see a dark band, okay, and a light band. The dark band, dark has an A in it, it's an A band. The light band, light has an I in it, it's an I band. That's not really where the A and the I come from, but that's the way you can easily remember it. Okay, so we have these dark A bands and these light I bands. Where these where these striations come from is from these myofibrils. They're lined up perfectly. They're lined up perfectly, so it gives the illusion of a stripe. There's no actual stripe there. Okay. So we're going to look more in depth at this myofibril to see where that repeating pattern is coming from. And where it's coming from is the... Autumn, can you put that away, please? Where that repeating pattern is coming from is it's coming from the sarcomeres, right? All of the sarcomeres being lined up. This sarcomere, right, is bounded on the edge here by what's called a Z-disc, and over here by a Z-disc. These blue lines are going to represent what we call the thin filaments. The thin filaments are actin. The pink line, or the red line, is representing the thick filaments, and those thick filaments are myosin. Yep, just head out of the room. You can always do that, just head out of the room. Okay. All right. So when we take a look at this, where we see that I band, that light I band, and that dark A band, the I band is when all of the different myofibrils have all of their sarcomeres lined up, and you have this kind of space where you don't see any of the thick filaments. Right? There are filaments there, but they're just the thin filaments, and so that appears to be a light band. Where the thick filaments are, that's where the A band would be. So when you have all of these within a muscle fiber lined up, you see those striations between dark light, dark light, dark light. Okay. Now this sarcomere itself, this is going to be the contractile unit. So if we look at this, now this is a three-dimensional structure. We're just going to take a little tiny section of it out and look at it. Okay. That Z-disc, that's kind of in between, right? It's in the middle. Okay. This is going to be where you have kind of an anchoring system. Right? Then we have overlap. Okay, there's overlap between these thin and these thick filaments. There is a M line, right, and that's right in the middle. Okay. That middle line is going to be where the contraction is going to move to. So in a simple sense, the way contraction occurs, we're going to look at something called the sliding filament model of contraction, but I'm going to kind of give you a sneak peek. The overall contraction in muscle is going to be due to an interaction between this thick filament and this filament, this thin filament. The thick filament is going to hook on to the thin filament, and it's going to pull it towards the middle. On the opposite side, we'll see the same thing happen. So this space is going to become smaller. Not by a huge amount. It's a tiny, tiny, tiny little amount of contraction. Okay? So this space in what's called the H zone is going to become smaller. The thin filaments are going to move towards the M line. And when you have this tiny little contraction occurring in a sarcomere, but then that sarcomere is repeated over and over and over and over and over again, you get a huge summation, and that's what actually gives you this gross muscle contraction. So in order to understand how that occurs, we need to look a little bit more in depth at these myofilaments. Right? What is actin? What is myosin? We'll start off with myosin. Okay. Myosin is going to be the moving molecule, right? This is the molecule that is going to use energy, and it's going to actually change its shape, and that's going to pull actin together. Okay. Now, if we talked about a single myosin molecule, it looks like a double-headed golf club to me. Okay. It has a rod-like tail, right? It's actually woven together. And then it has these two heads. Now again, this is just our way of representing the protein. Okay, remember what protein is. It's a chain of amino acids that has this complex organization, right? Secondary and tertiary organization. Right? Now the myosin filament is actually many of these molecules all together. And what that forms is a three-dimensional filament, right? So this would be our thick filament. Now you can see why it's called a thick filament, right? It's actually lots and lots of myosin all together. And what each of these heads do is what they have the ability to do is they can actually hook into actin, and they're going to be able to move the actin molecule. Right? The actin molecule, or the thin filament, actin is a more simple molecule. Okay, it's globular. Right? It's got a globular subunit. When all of those globular subunits are together, they form, basically it looks like a beaded necklace, right? Beads on a chain. And what this actin has is it has a special, what's called an active site. There's a spot on the actin. It's shown here as this little dark circle on each of the blue actin molecules, or the actin subunits. And that little active site is where these heads of the myosin molecule can fit in. Okay. Now there are two other proteins I haven't yet mentioned that are going to be important here. One is called tropomyosin, and the other is called troponin. And troponin is actually a complex. And these are located on the thin filament as well. And these are going to be proteins that act together. So troponin is usually shown as a complex of several molecules, and it acts almost like a bracket. And what that bracket does is it holds this other molecule called tropomyosin in place. And that tropomyosin is held in place, and it's blocking all of the active sites on actin. Okay? It's actually blocking the myosin from interacting with actin. These are considered regulatory subunits because these are going to regulate the interaction between actin and myosin. So if we put this together into a section of sarcomere, we now have this protein, this thick filament where all of these proteins are together. We have these myosin heads. These myosin heads want to interact with actin. Okay. The actin has on it active sites that want to be interacted with by the myosin heads. Normally, the actin molecules are blocked, their active sites are blocked by the troponin tropomyosin. Okay. What is going to happen in terms of how contraction occurs is we're going to see something called the sliding filament model of contraction. Okay. And in the sliding filament model of contraction, the thin filaments are going to slide over the thick ones, and they're going to actually shorten the distance between the two thin filaments within the sarcomere and make the entire sarcomere contract. Okay. The way that this is going to occur is by the myosin head binding with the actin, and then it's going to actually ratchet forward. Okay. When that happens, when it ratchets forwards, it's going to shorten the overall distance. Okay. So let's take a look back at this diagram here, right? And just imagine, if you will, for a moment. So the contraction that's going to occur within the sarcomere is really due to the interaction between the actin and the myosin. The regulatory subunits of troponin and tropomyosin are very important in regulating how that interaction occurs, because if tropomyosin is in place, you're not going to see interaction with myosin and actin. So what we need to talk about is a whole bunch of layers of contraction here. So the next thing we need to think about is, what are the other structures that are going to play a role here? Right? So we've been looking at this organelle that is made of these repeating sarcomeres. But there are other organelles within a muscle cell. Right? One of them is something called the sarcoplasmic reticulum. Right? Now that should sound familiar from our basic overview of cells. We talked about an endoplasmic reticulum. This is a specialized type of endoplasmic reticulum called a sarcoplasmic reticulum. Sarco meaning that it's particular for muscle cells. Okay. What this has an organization more like what you would see in a smooth endoplasmic reticulum, so we're not talking about ribosomes or proteins here. Okay. These are going to form channels that will go across the cell and into the cell and are going to connect all of those myofibrils. Okay. So this is going to be the system that's going to allow all of these sarcomeres within a single cell to connect at the same time, to contract at the same time. Okay. So if we look at just a section, okay, we're just going to pull a few of these myofibrils out of the larger cell. Okay. Running throughout this, connecting these all, we have what's shown here in blue. This is the sarcoplasmic reticulum. The sarcoplasmic reticulum is continuous with the sarcolemma through these things called T-tubules. Okay. These T-tubules are going to connect each of these myofibrils. This is going to be very important in the signaling. How do we get all of these sarcomeres to contract within a myofibril? How do we get all of these myofibrils within a muscle fiber to all contract at the same time? It's going to be due to these T-tubules and the sarcoplasmic reticulum. Okay. Now there is something that's referred to as the triad. Okay. The triad, tri meaning three. Here you have a central T-tubule, and on either side you have a part of this sarcoplasmic reticulum that is called a cisternae. Okay. This terminal cisterna, this is going to be where calcium ions are stored. We had previously mentioned the importance of calcium ions in muscle contraction. Now we're going to see what calcium ions actually do in muscle contraction. Okay. The calcium ions are going to be stored in these little pockets. Okay. The term cisterna, if you know what a cistern is, you can have a water cistern that collects rainwater that you can use then in your garden. Okay. That's basically this terminal cisterna. It's going to collect calcium ions. So this system is going to be acted upon by the nervous system. Right? right? And it's going to allow for the coordination of all of these different sarcomeres to contract at the same time. They are going to be continuous. The T-tubules are continuous with the sarcolemma, so they're going to have the same kind of organization as the sarcolemma, which also means that they are going to be susceptible to changes in membrane potential. Skeletal muscle cannot contract without input from the nervous system. And when we talk about the nervous system is a system of electricity. Right? So the ability to conduct that electricity is what's going to allow these muscle cells to contract. So as we try to put this information together, the first thing we need to think of, in order for skeletal muscle to contract, all skeletal muscle must be stimulated by a nerve ending. Now, we'll cover the nervous system in depth in chapters 11, 12, and 13, but in the simplest sense, nerve ending means that there is some neuron, right, some cell from the nervous system, and a signal is being sent from the brain to the muscle to contract. Okay, these are voluntary muscles, right? So when you want to contract biceps, your brain thinks contract biceps. Different parts of your brain send that signal to the muscle, and the muscle then contracts. So that's the stimulation by nerve ending part of it. Now, the electrical currents that are going to be involved in actually spreading the messages in the muscle cell are going to be called action potentials. So we're going to talk a little bit about action potentials in Chapter 9. I'm going to give you an overview of action potentials. And then in Chapter 11, we'll talk about action potentials in depth. There's going to be a key player in this, and that key player is going to be calcium. Now, I warn you, calcium is going to have two different jobs here. It's going to have a job on the neuron side of things, and it's going to have a job on the muscle side of things. So keep that straight. Okay. So this idea of when we talk about a nervous impulse, this electrical conduction, we call that excitation. Okay. So we're going to have a link between an electrical signal coming into the muscle and contraction, and we're going to call that excitation, contraction, coupling. In terms of how muscles are going to be stimulated, we're going to spend a good portion of Chapter 12, which is the central nervous system chapter, talking about the difference between motor neurons and what is the somatic nervous system. I put it out there now so you know what those terms are. That's basically the part of your nervous system that controls your voluntary movement, your body movement. When you see somatic means body. The important thing that we need to talk about here is the way signals travel in neurons. Right? The section of the neuron that sends the signal is called an axon. And that axon ends in motor neurons. It acts as something called a neuromuscular junction. And what a neuromuscular junction is, it's a junction between a neuron and a muscle. That neuromuscular junction, what's going to happen is going to be very important to this chapter. Okay? And a good portion of what you have to understand is what goes on at the neuromuscular junction. The neuromuscular junction is going to be where an electrical signal coming from a neuron is going to be changed into a chemical signal. That chemical signal is going to cross over a space, and then it's going to induce another electrical signal in the muscle cell. For the case of skeletal muscle and for what we will be learning, the chemical is what's called a neurotransmitter, and the only one we will occupy ourselves with in Chapter 9 is called acetylcholine. Okay? So here is acetylcholine. It is the neurotransmitter that's important at neuromuscular junctions. You can abbreviate ACH. Okay. This acetylcholine is going to be a chemical. That chemical is going to have the ability to bind to transmembrane proteins, which are ion channels. Okay. And it's going to allow those channels to open and close, and that's what's going to send on an electrical signal. Okay? So if you think back to chapter 3, when we looked at plasma membranes, we had those proteins that cross the plasma membrane that we said were ion channels, and some of them have to be bound with something. We call it ligand gating. And we'll spend more time looking at that in chapter 11 as well. All right, so let's see if we can give you the kind of short story to begin with. Okay, this is a depiction of a neuromuscular junction. Okay, this would be your neuron coming from the nervous system. Where it ends, we have what are called axonal terminals or axon terminals, terminal meaning the end. Where the axon terminal actually hits a muscle cell, now we're talking about a muscle fiber or a muscle cell, okay, we call that the motor end plate, and that's going to be an area that's a little bit thicker on the muscle cell. And these two things interaction, this is the neuromuscular junction. Right? So if we focus in on this, we're going to try and look at the steps involved in how a signal coming from a neuron can transmit across the membrane into the muscle cell. Now, these diagrams are slightly different than what you see in your text. These are from an older version of the text. They're not as pretty as the ones you have in your book, but I think they're more straightforward. Okay, so you actually will have two different versions that you can look at, right? The simpler version and then the more complex one. All right, so here's the short version of the story. Okay. When we talk about electrical impulses, they are something called an action potential. We will mention what that is, but for now, just take it as like hand waving magic. It's this thing called an action potential. It's a change in the membrane potential. It's a signal. That's how neurons send signals. That's how muscles are going to send signals. Okay. So that action potential signal is going to come down from the brain or wherever it originates through the neuron here into this axon terminal. Now this is the beginning of that excitation-contraction coupling. When this signal gets here, the first thing that it's going to do is it's going to open a door for calcium ions. Okay. Now it's not really a door; it's a membrane. It's a membrane channel, right? A protein that's in the membrane. Okay. So it's going to open up a channel for calcium. Right? That calcium ion is going to come in. When this calcium ion comes into this axon terminal, it is going to make its way down to these vesicles. These are neurotransmitter vesicles. Remember that vesicles are basically little bubbles of plasma membrane within a cell, and when that cell makes something, it can package it into vesicles. These vesicles are going to be full of acetylcholine. What calcium does is it makes these vesicles move to the very edge of the cell and exocytose. Right? Exocytose meaning they're being released into the space between the neuron and the muscle. Okay? That space is called a synapse. So we have an electrical signal that comes in, and here's where it's going to change to a chemical signal. Okay? And there's a reason for that. Electrical signals can't jump over open space. Right? So the electrical signal coming in here is really due to changes in the plasma membrane. But there's no plasma membrane in a space. So we can't have that same kind of signal go from here to here. There's got to be a different form to get it across the space. Now when this acetylcholine gets released, right, it's going to make its way across that space, that synapse, to the plasma membrane of the muscle cell. Okay. Now if we focus in on this last part, this is showing us a close-up of the exocytosed acetylcholine. So these kind of green circles are representing acetylcholine. They're going to be released, and when they get released, they're going to going to bind to transmembrane proteins here, and they're going to open doors for other ions. Okay, and those specific ions are going to be sodium and potassium. Now, when I say open doors, again, these are really proteins that they're going to open. We call these ion channels. Now, what that does, opening these channels, what that does, that's going to cause another action potential. it leads to something called depolarization, and then leads to another action potential. So we have an action potential that comes in through the neuron, changes to a chemical signal through a neurotransmitter, and then that neurotransmitter, that chemical, causes another action potential. Right? That action potential can then move all along the sarcolemma. It's going to happen all along here. We're just looking at one point, and it's going to travel down the T-tubule. Right? Once it travels down the T-tubule, the thing with these electrical signals, these action potentials, is they are fast. Once they start, they don't stop, and they go really, really fast. Okay? So that signal is going to spread out in all directions at once, and it's going to go all down all of the T-tubules. Now, what that action potential is going to do is it's going to be the signal that's going to kick off contraction. All right, so when this occurs at the neuromuscular junction, right, it's going to initiate an action potential that's now going to pass through down the T-tubules, okay? And when it passes down the T-tubules, that action potential is also going to be able to open calcium channels, but now we're talking about calcium inside the muscle. Previously, we had seen that up here in the axonal terminal, action potential can lead to calcium coming in the neuron. Well, as that depolarization passes down the T-tubules, it can make that calcium channels open, and now what's going to happen is the calcium that was in storage in those cisternae, in the sarcoplasmic reticulum, it's going to leave the cisternae and it's going to go flooding out into the cell, right? So it's within the cell, right? It's not in storage anymore. It's now free to go and interact. And what that calcium ion is going to do, here's the second job of the calcium ion. It's going to make its way over to all of the sarcomeres. Now, we're just looking at one tiny little part. We're not looking at all the sarcomeres. But this happens at all of the sarcomeres. And what that calcium is going to do is it's going to bind to troponin. Troponin was one of those regulatory subunits. Okay? And it's going to bind to troponin, and it's going to make troponin change shape. And when troponin changes shape, it's going to pull back tropomyosin. And when it pulls back tropomyosin, it's basically going to allow the myosin heads to interact with the actin molecules. The only thing that keeps myosin from interacting with actin is the fact that the actin active sites are covered by that tropomyosin. So as soon as calcium comes and changes the shape of tropomyosin, that tropomyosin basically pulls back and pulls the tropomyosin out of place. Now myosin is ready to go. As long as there's ATP present, which we assume if a cell is alive, there's ATP present, that myosin then is going to come, it's going to hook into actin, and we're going to start seeing that sliding filament model of contraction. And as long as calcium is present, you're going to have that ratcheting occur, and you're going to have the contraction occur. Now, the way this stops, the way this turns off, is if there's no more signal coming here, right? There's no more signal coming in through the neuron, then there's no more acetylcholine being released. Once acetylcholine binds, it doesn't stay bound forever. It gets taken up again or it gets broken down. So if there's no more signal here through the neuron, if there's no more signal here in the neuron, and then there's no more acetylcholine means no more binding of those ion channels here, which means no more depolarization or action potential here, which means no more action potential here. If there's no more action potential, these calcium doors are going to close, and that calcium is all going to get sucked back up into the cisternae. When this calcium gets sucked back up into the cisternae, it's not bound to troponin anymore. And troponin goes back to its resting state, and it basically slams that tropomyosin back into place, covering up the actin binding sites. And now myosin can't get to actin anymore. Okay? And that's what's going to allow the myosin to disengage from actin, and those actin are going to slide back to rest. Okay? So a contraction, in order for a contraction to happen, you have to have calcium present, right? So the neuron sends a signal in the form of action potential, and we'll talk on Monday about what action potential is. Neuron sends a signal in form of action potential. The signal crosses the synapse in the form of acetylcholine. Acetylcholine binds to ion channels, and that's going to kick off another depolarization, which leads to another action potential. That action potential is going to travel down the T-tubules. Remember, those T-tubules wrap all throughout the entirety of the muscle cell. So they all get this very quick signal. They all release the calcium, goes over to the troponin. Troponin changes shape. Tropomyosin gets pulled out of the way. Myosin is ready to go, starts ratcheting in, and we have the contraction. When there's no more signal, the calcium goes away and relaxation occurs. Now, this is kind of the broad strokes picture of this. There are a lot of fine details that I need to fill in, and we'll fill those in on Monday. So, in terms of talking about how this muscle contraction occurs, an important component is action potential. And action potential, the definition of an action potential is a transient depolarization event that includes a polarity reversal of the sarcolemma or the nerve cell membrane and propagation of action potential along the membrane. So let's explain what that means. Okay? Whenever we talk about a membrane potential, okay, potential means a difference. In Chapter 2, when we looked at energy, we talked about potential energy and kinetic energy. And I said that if you have a potential, it's because you have two things that are different, right? If you hold a ball up in the air, and it's got potential energy, right? If you drop it, then it's got kinetic energy, right? So potential energy is a way of doing things. Specifically, a membrane potential is when you have a difference in the number of ions across a membrane. This is covered in Chapter 3. There's a subsection that gives you an overview of membrane potential. So if you haven't looked that over, go back to Chapter 3 and review that. Right? But in a simple sense, all plasma membranes are similar in that they're made of phospholipids. So because they are basically lipid in the center, things that are charged cannot pass through. Right? That was something we established way back in Chapter 3. So things like ions, sodium ion, potassium ion, calcium ion, chloride ion, these things have a charge to them. They are ions. So therefore, they cannot pass through the plasma membrane unaided. Okay. They can pass through the plasma membrane if there are ion channels. And we did look at ion channels, again, back in Chapter 3, looking at the cell. What is going to be unique about muscle cells and neurons is we'll see that those cells have a way of establishing a difference, right, a potential, a gradient across their plasma membrane so that they have more of one ion outside than inside, more of a different ion inside than outside. And we're going to spend a good deal of time talking about the specifics of that in Chapter 11. Now, when you have a potential like that, if you have a store of a lot of stuff outside of the cell and very little on the inside, there's a desire, right? A movement, right? And a desire to move into the cell. Okay? So if a channel is there and that channel gets opened, if you have a lot of, say, sodium outside of a cell, very little inside a cell, we say it's a concentration gradient. Now, if there's no channels, that can't move anywhere. But if we put a channel in there and we open and close selectively that channel, we can control how that sodium moves in. That's going to be the key thing to looking at an action potential. Now, when we use the term depolarization, okay, when you have something that is different, right, it's polarized. Okay? If you have something that's got more positive outside, more positive inside, we say that it's polarized. Right? You can use the term polarized for a lot of things. Right? In terms of politics, you might hear that term. If you do like a poll of people, you might find what are called polarizing topics. Right? Big things are things like abortion. Big things are like immigration reform. Right? Most people have really strong feelings one way or the other. So they're either like yes, 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 or no, no, no. That's called a polarizing topic because you get two very different aspects. So when you have something that's polarized, you can imagine that they are some distance away from each other, whether it be a physical distance or some other like distance of ideology or a distance of charge. Okay? And that's what we see when we talk about muscles and neurons. A depolarization then means making them less different. Right? So they start to become more similar. So that's what the depolarization part means. And we'll look at the specifics of how that happens a bit more in Chapter 11. Okay. Now the term transient means something that comes and goes. Okay. So what we're going to see in terms of an action potential is that we're going to change the polarity of this muscle cell, right, the sarcolemma, right? But we're going to do it, it's going to come and go. Right? It's transient, it changes. We'll also have a polarity reversal. What that means, if you have more positive outside than inside, when you depolarize, those two things become more similar until they actually flip-flop or they switch. And then you get more positive inside than outside. Okay, that's a reversal. Now what that's going to do, that is going to allow for a signal to be spread along a plasma membrane very quickly. And that's what we'll see in both muscles and neurons. In terms of the neuromuscular junction, what's going to kick this off on the muscle side of things will be the binding of acetylcholine. Acetylcholine is a neurotransmitter. It gets released from a neuron. It crosses the synaptic cleft, and it's going to bind to what's called a ligand-gated or a chemically gated channel. When it binds, it will open that channel, and we are going to see that we'll see movement of sodium ion and potassium ion. When we see that movement of sodium and potassium, we're going to change the resting membrane potential of the muscle cell, and we're going to lead to a depolarization. So we're going to see that that potential, that difference from the outside to the inside, is going to get less different. Okay? That getting less different is going to kick off that action potential. Now this starts out locally, right? So this initial change starts out just around those chemically gated channels, right? So when acetylcholine binds, it opens a channel, sodium is going to come in, potassium is going to come out, and that's going to have an effect, a depolarizing effect, just around that channel. Right? So we can't have this happen if we just have one channel opening. We have to have lots of those channels opening. And if enough of those channels open, that's going to lead to the action potential. The action potential then can spread, right? It's no longer localized. And we'll see why that is when we look at the neuron, but I'll give you a hint. It has to do with ligand-gated versus voltage-gated channels, right? And we'll talk about what that is in Chapter 11. So here's kind of an overview of action potential. Right? If we imagine that this is a very simplified part of a muscle cell. We've taken out all of the other stuff. We're just really looking at the sarcolemma. Okay. Now there's lots of different ions, right? But overall what we see, we're only going to talk about the two that are important for right now, and those are sodium and potassium ions. Okay. The outside portion over here, we see that we have relatively more sodium outside than inside. When you have these little brackets imply concentration of. Okay, so this basically tells you have a higher concentration of sodium outside of the muscle cell than inside of the muscle cell. Right? In the opposite way, we have relatively higher amounts of potassium ion inside than outside. Okay. Now what that leads to a relative difference here. Now this is a relative difference. So if you compare right along the plasma membrane, you have relatively more positive outside and relatively less positive inside. And you can also think of less positive as being negative. At rest, we call this a membrane potential. You can look at this and you can see how there's potential there. Right? There's potential because if given the opportunity, sodium's going to go in, right? Because there's more of it outside than inside, it's going to go in. If given the opportunity, potassium's going to go out because there's more of it inside than outside. That is potential. Right? That's what we call potential. Think again about holding up a baseball. If given the opportunity, if you let it go, it's going to fall. It's going to have energy. It's going to do something. It's going to move. Now, for the most part, we'll see that the sarcolemma, the plasma membrane of the muscle cell, is impermeable to both of these ions. So the ions cannot pass through willy-nilly. They're only going to be able to pass through certain types of channels. Okay? They have to have a protein that's embedded in the plasma membrane so they can pass through. Now, the first kind of step involved here is that localized depolarization. Right? When you have that axonal terminal, right, of a motor neuron, that neuromuscular junction, releasing acetylcholine. When that acetylcholine is released, it's going to cause a part, right, a little region of the sarcolemma to become permeable to sodium. Right? And when we say become permeable to sodium, it means sodium channels are opening in that area. When sodium channels open, when you open a door from sodium, it's going to want to flow in. Okay? The reason it wants to flow in is because there was more sodium outside than inside, so it's going to go along its concentration gradient. Now, what you have to think about happening here is we don't actually have pluses out here and minuses inside. It's a relative distance. It's a relative difference. So as sodium starts going in, the outside is going to become less and less positive, and the inside is going to become more and more positive as they approach one another. And that's where we get our depolarization. Okay? You're losing positives from the outside, and those are being gained on the inside. So we're seeing that the difference between them is going to become smaller and smaller. Now, we say that as that sodium is entering the cell, the resting potential is going to be decreased. We get that depolarization. Now, if that stimulus is strong enough, right, if there's enough acetylcholine opening enough channels and enough sodium comes in, it's going to kick off an action potential. Right? Now, notice here we have action potential versus resting potential. Right? A resting potential is the potential of the sarcolemma, or later on a neuron, when it's not doing anything, when it's at rest. Now, think about it. When you are at rest, it's not that nothing is happening, right? But there's no big movement. Okay, your heart's still beating. Your cells are still doing things. So when we talk about a neuron that's at rest, or we talk about a muscle cell that's at rest, there are still things going on, but it's not doing its job, right? We're not going to see a neuron sending a signal at rest. We're not going to see a muscle contracting at rest. But there are other things that go on at rest. Rest does not mean that nothing is going on. So if there's enough of a signal coming from acetylcholine binding, enough sodium coming in, enough of a depolarization, we're going to switch to an action potential. Right? And so now we're going to see the cell in action doing what that cell does. So in the case of a muscle cell, it's going to lead to contraction. Okay. Now, the way that action potential is propagated, right? When you propagate something, you pass it along. You send it along. Okay. So we can then say that action potential is propagated or sent down the sarcolemma and then eventually sent down the T-tubules. The way that this occurs is when you make a change here, that change is going to induce a change in the next patch, and that's going to be due to those voltage-gated channels. Excuse me. At the beginning of this, the depolarization is due to the acetylcholine binding to ligand-gated channels. Okay? But there are also channels that we'll see that are sodium and potassium channels that open due to a change in membrane potential. Okay? Those are called voltage-regulated or voltage-gated channels. So once we start off this depolarization, if that depolarization is big enough, it's going to start opening the adjacent channels. And then that's going to lead to a depolarization. And if that's big enough, that's going to open the next ones. Right? And if that's big enough, it's going to open the next, and the next, and the next, and the next. Right? Once this happens, it becomes self-propagating. So it's like if you play dominoes, not the game dominoes, but line the dominoes up. If you knock that first one down and they're set up well enough, they're all going to go down. Boom, 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 down the line. And that's basically how action potential is going to get propagated. This happens very rapidly. Once this happens, you're going to see that this is unstoppable. Okay. So you don't have an action potential that stops. Okay. So once that signal comes in from the neuron, it's going to continue. What this will eventually lead to, again, we have this action potential is going to be a means to send a signal down the T tubules to open those calcium channels. Calcium then is going to go and bind to troponin. Right? So we're just kind of filling in the blanks on what is this action potential here. Now you can imagine if action potential just kept going, well, things have to get set up again. Okay. If you imagine a string of dominoes, you knock one domino down and they all fall down, well, you can't send another signal till you set all those dominoes up again. That's going to be really important. We have to set this up again. We have to bring the sarcolemma back to resting potential so that way the next contraction can come through. Right? Otherwise, the muscles would basically stay contracted. So once this wave of depolarization passes, okay, we have the reversal of membrane polarity. So this is the area that's depolarized. It actually has the reversal. Now we have positive, more positive on the inside than the outside, right? Not shown here, but you can imagine there's now more sodium on the inside because that sodium had come rushing in. We now need to flip this back. We need to get back to that right amount of polarization. So this is where the calcium is, this is where the potassium is going to come into play. Okay. Here we're looking at the interaction between sodium and potassium. When sodium stops moving, now we're going to start moving potassium out. Potassium is also a positive ion. So as potassium starts moving out, we're going to see repolarization. Okay. So repolarization means we're going back to that resting membrane potential. The inside versus the outside is going to become more different again. When this occurs, then, we reestablish that resting membrane potential. That repolarization gets us back to this point of having more positives outside than inside, having this relative positive measure outside compared to inside. But where are the ions? Okay. If we think about this just repolarization occurring, now we have lots of potassium outside, lots of sodium inside. Right? That's not what we want. In order to start this whole thing over again, we need lots of sodium outside compared to inside, lots of potassium inside compared to outside. So that's where we're going to bring up the sodium-potassium pump, or the sodium-potassium ATPase, that we looked at way back in Chapter 3. We talked about active transport. This pump is always running in the background. Remember I said at rest there is stuff going on? This is one of the things that's always going on. The sodium-potassium pump is a pump. If you don't remember what it is, go back to Chapter 3. We looked at the diagrams. What that pump does is it actively pumps sodium out and pumps potassium in to get us back to having that correct resting membrane ionic concentration. So we go back to having more sodium outside than inside and more potassium inside than outside. So there's really two things that get us back to the start. We have repolarization, which is the change, and then we have the sodium-potassium pump. Now this all happens, it takes me minutes to describe it to you, but it doesn't take minutes, obviously, for this to happen. Okay? This happens extremely quickly. We're talking about the movement of ions over really, really small distances. We're talking about fairly small numbers of ions that are moving. Okay? And we'll look at a little bit more at the concentration. Now, another way to look at this action potential is to look at it in a term like this. This is what's called a scan. Okay? When we are talking about membrane potential, it's actually something that is measurable. Okay? It's measured in millivolts, okay, which is 1/1000th of a volt is a millivolt. Okay? When we talk about volts or voltage, that's how we measure electricity. If you do any electric work and you want to find out if something is working, right? You have these little metal things you can touch to it, and it's got a little voltmeter. Okay? So you can imagine we could take a little tiny, teeny voltmeter. Okay? And they actually do have these. These are made of extremely thin glass rods, and you could actually touch a cell, and you could measure the voltage there. Right? It's measured in millivolts. Now we're going to give you some general numbers depending on what text you read. There's a range here, but I'm not going to try and get you to remember a range. So I'm going to give you a set of numbers that I want you to know for this class, and it serves as a good basis for how membrane potential works. Right. So in terms of if we have a cell at rest, resting membrane potential we usually can think of as being minus 70 millivolts. Now this minus 70 millivolts, what this means, this is a relative measurement. So if we took a little tiny voltmeter and we measured the outside and the inside, we'd see that the inside is basically 70 less than the outside. That's with the negative 70. That's how we get a single number. We're talking about outside versus inside. Okay. So the inside is like 70 less. That is our resting membrane potential. It means that if we have 70 less on the inside, it means we have more positive on the outside. Now the way an action potential is going to occur, that first part, that first depolarization, in the case of a muscle cell, when we have acetylcholine binding to ligand-gated channels, we have that localized depolarization, that's going to lead to some small amount of depolarization. If there is enough of that small depolarization that's localized, it may hit something called threshold. Now, threshold is going to be extremely important in neurons. Basically, what that threshold is, it's an amount of depolarization that is necessary to open voltage-gated channels, right, to kick off action potential. Okay. So in the case of neurons, and we'll kind of use the same relative number for muscle cells, we use minus 55 millivolts, right? So the difference from minus 70 to minus 55, okay, what we have happening here is you can imagine, oops, okay, if you have minus 70 and positive sodium starts coming into the cell, this minus number becomes less and less minus. Okay? So minus 70, if you have negative $70 in your checking account, and I give you $15 to put in your checking account, you'll now have negative $55 in your checking account. Okay. So what's happened here, getting up to this threshold, that's going to be due to that depolarization, usually due to ligand-gated channels. And we'll see that it's going to be the same for both muscles and neurons. Now, once threshold is hit, that's when we're going to see the action potential kicking in, because now the channels that are going to open are going to be those voltage-gated channels. When you have a voltage-gated channel that opens, okay, it's going to cause a depolarization that's going to be big enough to open the next and the next and the next and the next, and that's where we get that propagation. Okay. So what we have is we have a lot of sodium channels opening very rapidly, and as we have lots and lots of sodium flowing in, we see this huge, very fast depolarization. Okay. So now we're going from minus 55 all the way up here at 0. At 0, the inside and the outside are the same in terms of their charge. Then we have this movement from 0 up to plus 30. This is where we have that reversal of membrane polarity. So now the inside is actually positive relative to the outside. Okay. This depolarization, okay, is going to occur very quickly. Now at some point, we're going to see a change here. And we usually put that change point at plus 30. Right? And at plus 30, two things are going to happen. The first thing that's going to happen is sodium channels are going to close. When sodium channels close, we're also going to see potassium channels open. Right? So that means no more sodium is coming into the cell. And now potassium can start going out of the cell. And when potassium starts going out of the cell, the inside of the cell, which is now relatively positive, okay, plus 30, is going to start becoming less and less positive, and we're going to see repolarization occurring. Okay, we're going to go back down towards resting potential. Now this also shows an area here called hyperpolarization, where the membrane potential is actually lower than resting potential, but we're going to kind of just put a pin in that for now. We're going to see the same action potential scan when we look at neurons and see it more in depth. This is just another way to visualize what's going on. Okay? If you're good with numbers and numbers help you to understand things, this is a good way to think about it. So on this, the blue line is what you measure, right? We use this axis here, membrane potential. We have things being measured in millivolts. Okay? But there's also this orange line and this green line. The orange line and the green line are measured off of this axis. So this is a really weird scan. It's got two different axes, two different y-axes. This is relative membrane permeability. And previously what I said was when you talk about membrane permeability, it's really a measure of how many channels are open for a particular ion. Okay. So this orange line is going to be the permeability for sodium. Okay. So you can see that when depolarization hits threshold, right? If we look at time, if you follow this straight down and see time, when that threshold happens, boom, you're going to have a huge increase in membrane permeability for sodium. That means lots and lots of sodium channels are opening. Okay. Then here, where the sodium channels close, right, where we see this drop off going from depolarization to repolarization, we're going to see a huge decrease in the membrane permeability for sodium. That means the sodium channels are closing. This greenish line, this is for potassium, so same thing for potassium. You notice that if we follow in time, right at this white line here, okay, here's where you have a big increase in potassium channels opening, and then they actually close slower, and that's going to be very important when we look at neurons. So just a way for you to kind of look at these three interacting lines at the same time to see what they're doing. Please notice the time scale down here. This is time in milliseconds. This is 1/1000th of a second. So we're talking about this entire action potential taking 4 milliseconds. Okay. So within a second, you could theoretically have 250 action potentials going through something in a second. 1/1000. Questions on this action potential scan? We're going to see it again. We're going to see it in Chapter 11. We're going to talk about it more. All right, so let's remember, why are we even talking about action potential? Why are we talking about action potential in muscle cells? The reason for this is the action potential is what's going to allow for there to be the excitation-contraction coupling. Okay. Remember the excitation, that is that electrical signaling. The contraction is the interaction between actin and myosin, and putting them together is the coupling. So once we make this action potential, that action potential, it's going to spread along the sarcolemma rapidly in all directions, and it's going to travel down the T-tubules. When it travels down the T-tubules, it will release, or it will lead to the release of calcium from the terminal cisternae in the sarcoplasmic reticulum. And the way that it does that is that action potential can affect voltage-gated channels or voltage-sensitive channels. So as that action potential passes, it's going to open those channels and it's going to release calcium. And that calcium is going to move to the sarcomeres where it's going to bind to troponin. Okay. When it binds to troponin, it will change the shape of the troponin, and it's going to actually move that regulatory subunit of tropomyosin off of the actin binding sites. That's going to allow the myosin to form cross bridges where the myosin head actually interacts up and into the actin. And we'll see that the thin filaments are going to move towards the center, and that's going to shorten the sarcomere. Right. ATP is important here. Right. So that's how we have that excitation-contraction coupling. In order to stop this whole process, calcium has to be taken off of the troponin, taken back into the sarcoplasmic reticulum. As soon as calcium comes off of the troponin, troponin goes back to its resting state, puts tropomyosin back in place, and then myosin can't interact, and that's when relaxation will occur. Way back the first time we talked about this, we said muscles have the ability to contract, relax, and extend. So when a muscle is not actively being contracted, it's going to go back to its resting position. So let's just take a little bit of a closer look at how calcium does this in terms of how calcium binds to troponin. Now what we are looking at here, this would be if you were to take a cross section of a thin filament right at the place where we have the troponin holding on to the tropomyosin. Okay, so this would be the actin subunits. Here are the myosin binding sites. This is the troponin complex. Okay, and that's holding the tropomyosin in place. And there's the tropomyosin right there. That tropomyosin is preventing this myosin head down here from getting into the actin binding site. So at rest, when we do not have contraction, the troponin is in this kind of orientation. And again, we are using blue circles and yellow circles to represent very complex protein shapes. Okay. When calcium ions bind to one of the subunits of troponin, it's going to start changing the three-dimensional configuration of the troponin, and the troponin is going to fold back on itself, and when that happens, it's going to start rolling the tropomyosin out of the way. So you can see this was the initial position of the tropomyosin blocking the active site. Now it's being moved out of the way. When that occurs, now this myosin is going to be able to interact with the active site. Okay. Once this is able to interact, as long as there's ATP present, once this is able to interact, we're going to see that this myosin can now go through its cross-bridge cycle, power strokes, and actually lead to the contraction of the sarcomere. Now that what myosin does, you can think of it as 4 steps. Again, you don't have to memorize which step number 1, what's step number 2. This is a cycle. So the term cross-bridge formation, this is when the myosin is going to actually make what's called a cross-bridge. It's crossing or bridging, okay? And that's when it attaches to the actin filament. Okay? When there is the working stroke or the power stroke, that's when the myosin head is going to pivot in and is actually going to pull the actin towards the center. Remember, we said that the center of the sarcomere is called the M line, right? The middle line. Once that ratcheting has occurred, then the myosin is going to release and it's going to take in new ATP, and that's going to lead to that myosin getting cocked and ready to go. Okay. Now that cocking is the same way you cock a gun. It's got the energy now. It's going to split that ATP. It's called hydrolysis. It's not going to have the energy. If actin is still available, this cycle will go again. Okay. This cycle will continue as long as ATP is there, and as long as actin is exposed, tropomyosin's out of the way, this myosin is going to go through this cycle. Now we can look at this visually. Okay. Here we have our actin subunits. This would be our troponin. Here is our tropomyosin. What is going on here is we are assuming that there is calcium that has been released through excitation, contraction, coupling, that calcium is bound to troponin. Troponin has changed shape. Tropomyosin is moved out of the way. Okay. When the myosin head is in its high energy configuration, it is interacting here. It is holding on to this ADP and PI. Okay. What that means is it's taken in an ATP and it's broken the ATP and it's holding on to the energy. Okay. That energy is really in the form of that PI. That's the inorganic phosphate. That energy, my mouse keeps disappearing. That energy is going to then go into moving this myosin head. So it's going to ratchet forward, and that's going to start bringing those thin filaments closer together. Once that occurs, then, we're going to see the release of that ADP, and a new ATP can come into place. Right? When that ATP comes into place, we get the myosin letting go of the actin. It then is going to break that and release the energy, get the energy into it. Okay? That's going to get it cocked, or it's going to get it ready to go. And then it's going to hook up again. And this cycle is going to continue and continue as long as there's ATP and as long as actin is exposed. Okay. That's what allows us to have sustained contraction. Okay. As soon as this stops, as soon as calcium is taken off, as soon as troponin goes back to its resting state, as soon as tropomyosin blocks this, myosin has to let go, and that's when we have relaxation. Now this control, in order for this to work, this control is really important. Okay? A big part of what we will talk about in chapter 11, 12, and 13 is how do the neurons interact with the muscle system. Okay. Salute. So what we need to talk about here, just very briefly, because we haven't really gone into the nerves, we haven't gone into neurons too much, is what's called the nerve muscle functional unit, or the motor unit. A neuron, which is a cell of the nervous system, is going to supply signals to possibly one, possibly more muscle fibers. Remember, muscle fiber means muscle cells. Now, depending on how many cells a single neuron controls, we can see a different level of fine-tuning of our control of contraction. Okay? So if we have muscles that have to do really fine movements, right? Remember now, we're talking about muscle as the organ made of many, many cells. Okay? If you have a muscle that is made of many, many cells, and it has many neurons, each talking to a very small number of cells, you can control that muscle much better, right? Because you can contract this little bit, or that little bit, or this little bit, or that little bit. Okay? So let's say to have a very small motor unit. As opposed to muscles that have large motor units, a large motor unit means one neuron talks to a lot of muscle cells at the same time. Okay? When you talk to lots of muscle cells at the same time, you have less fine control, right? Imagine if when they built this building that we're in, they only put in one light switch. You walk in the front door of the building, you turn on all the lights in the building. Right? Does that make sense? No. Okay. But if we had that in maybe a much smaller building, or in a building where we want all the lights to come on at one time and off at one time, that might be the right way to do it. Okay. Here we have light switches for each room, and in fact each room has 2 light switches. Okay. I would prefer if there were 4 sets of light switches in this room. Okay. You can imagine if we were in a photo studio. Okay, this room was a photo studio. We'd have a lot more light switches because we might want just that light on, or just that light on, or just that light on. Okay, that's basically what we're talking about in terms of these motor units. Okay, so if you take a look at just a kind of general depiction of this, we can see here 2 different motor units: motor unit 1 and motor unit 2. Motor unit 1 is shown in purple. Motor unit 2 is shown in red. Motor unit 1, this neuron branches and controls this muscle cell and this muscle cell. Now you cannot control, when we talk about neurons either send a signal or they don't send a signal. Right? You can't send a signal to only part of a neuron. Okay? So when this neuron fires, it's going to contract both of those muscle cells. Here we have a motor unit that controls 1, 2, 3. A good example of this is, think about your facial muscles versus your quadriceps, or your thigh muscle. Okay? Facial muscles are going to have very small motor units because you can do really, really, really tiny movements of your facial muscles, right? You can contract a little bit more, I mean, until you can get to like Joker smile contraction. Okay? That's because we have very small motor units. So there are many more neurons or much fewer muscle cells per neuron in your facial muscles, as opposed to, say, your quadriceps. Quadriceps, really, you could pretty much contract them or relax them. Contract them or relax them. Most people don't have really fine muscle movement. Okay? Most of our big muscles have fairly large motor units, where it's contract it or relax it. Contract it or relax it. Whereas with the facial muscles, things like the hands, the reason we have all the dexterity we have in our hands, the muscles that move our hands have much more dexterity because they have much smaller motor units. Does that make sense? Okay, good. All right. So in terms of smooth muscle, this is the muscle we would consider involuntary muscle. It is what you would see in your hollow organs. You'd see it in some of the blood vessels. This does not have the same organization that we would see in skeletal muscle. We do not see striations in it, hence the reason it's called smooth muscle. The cells themselves, the fibers themselves, are spindle-shaped, so they are fatter in the center and thinner on the edges, as opposed to being rod-shaped like skeletal muscle. We also do not see those striations, which means we don't have that same arrangement of sarcomeres. Right? Skeletal muscle has those striations because of the way the sarcomeres are attached. We also are not going to see the bundling of fascicles in the organ. Right? We're not going to see that parallel arrangement. What you do have a fine endomysium, which is that innermost layer of connective tissue, but we're not going to see the other connective tissues. When we have these lining the hollow organs, all of the hollow organs except for the heart are lined with this. Right? It's going to allow for contraction. Think about the esophagus. I think the esophagus is a really good one to think about because we all can feel what the esophagus does when we swallow. Right? It moves stuff down into our stomach. Right? One of the ways that it does this is the smooth muscle within the esophagus is organized into two layers. Okay? It's organized into a longitudinal layer and a circular layer. The circular layer basically goes around the circumference of that organ. The longitudinal runs the length of that organ. Now, they have a very similar contractile mechanism as skeletal muscle, obviously without the sarcomeres. So we are going to see calcium playing a role. We're not actually going to go in depth into the mechanism of smooth muscle. Okay? We're going to focus on the skeletal muscle. If we were to take a look at a section, this is a section coming out of the small intestine. Okay. The small intestine has, of course, it's an organ, so there are multiple kinds of tissues here. The very inner part of the small intestine, this is called the lumen or the space. Okay. This is going to be lined with epithelial tissue. Okay, that epithelial tissue is there because that's where we have absorption and secretion and things like that going on. That will sit on a layer of connective tissue because epithelial tissue always sits on connective tissue. Then we have two layers of muscle, and on the outside, then we have some more connective tissue. So this is really what we want to concern ourselves with, these two layers. The circular layer, which runs around the circumference, and it is circular, and the longitudinal layer, which runs the length of the organ. Now, depending on how we cut through this, right, if we cut through this, the smooth layer, the circular layer of smooth muscle, you could really clearly see the actual spindle shapes of the cells. Whereas if you cut through the longitudinal layer, you're cutting all of these cells in cross-section. And because of the way they're arranged, you see them looking all different sizes. The key thing when you are looking, and when we look at this in lab, you will see that the key thing is that you see two layers here. That's your hint that you're looking at smooth muscle. You don't see that same kind of layering when you have skeletal muscle. Right? The other thing is it's smooth. You don't see any striations. Right. Now, the way this is going to work, when you contract the long layer or the longitudinal layer, the organ will dilate and contract. When you are going to contract the circular layer, you're going to see that you're going to have the tube elongate. Now, the best way to think about this, right, is what happens in what's called peristalsis. Peristalsis is going to be a alternation of contractions and relaxations of the smooth muscle, right, that are going to squeeze substances through the lumen, right? So this can happen in the esophagus, this can happen in the small intestine, right? So I like to think about like a tube of toothpaste, right? If you squeeze the tube of toothpaste and then pull your fingers up it, you're going to move that toothpaste out of the tube, okay? And that's basically what happens with peristalsis, okay? You're going to move a substance through the tube. When you swallow something and it passes through your esophagus, it doesn't fall into your stomach through gravity. Okay? We see this alternate contraction, relaxation that actually is going to squeeze the food downwards. Same thing has to happen through all of your digestive system to keep food moving in the proper direction. Okay? So that's going to be an important component of smooth muscle. Now, smooth muscle is going to be innervated differently because we are not going to see conscious control. You cannot consciously control what's going on in your smooth intestine right now. Therefore, the part of the nervous system is going to be different. It's going to come from something called the autonomic nervous system, which we'll talk all about in Chapter 13. And we have a slight different organization. So we don't see a neuromuscular junction the way we saw in skeletal muscle. We have something that's similar, and those are called varicosities. Okay? The varicosities are going to release neurotransmitters onto synaptic clefts, but here these are bigger. They're called diffuse junctions. So if we take a look at this, okay, here is the same section of lumen of the small intestine. Here's the muscle we're talking about. Now we have what's called an autonomic nerve fiber. Okay, so this is not somatic, this is not body control, this is not voluntary. Okay. Notice that the swellings are not necessarily at the end of the neuron, they are actually along the neuron, and these are called varicosities. What happens in the varicosity is actually very similar to what happens in the neuromuscular junction, in that there are synaptic vesicles, and those synaptic vesicles are going to release their neurotransmitter. Now, there are different neurotransmitters here, okay, depending on what we're looking at, so we won't go through all of them. But the process then, that's going to kind of kick off the process as well. So this is how the neuron communicates with the smooth muscle, okay, different organization. Now, the way that a smooth muscle cell is going to contract, because we don't have that rod-like shape, we don't have those myofibrils, we don't have actin and myosin in those straight sarcomere bundles. Instead, what we have are what are called intermediate filaments, all right, and they're attached throughout to these things called dense bodies. Right? When a smooth muscle cell is relaxed, it's elongated. When it contracts, it's going to forcibly shorten. Okay. The best way I can describe you to think about this is if you've ever watched how a slug moves, right? Slug can extend out and then it can contract, and that allows it to move. Right? Now, in our bodies, for the most part, we don't have single cells of smooth muscle. Right? These occur in sheets. They occur in a circular layer, longitudinal layer. So we don't have a single cell that's contracting. We have the whole thing contracting. When the entire sheet contracts, that's when we get contraction around the lumen, right, where we actually make that smaller. And so just to conclude this, there's a nice overview in your book and in your notes that compares skeletal, cardiac, and smooth muscle. Okay. You do need to know the location and the examples and the microscopic organization of all three. Okay, so this first part of the table, you need to know it all. Okay, where is it? How is it controlled? What does it look like under the microscope? We've seen that before. That should be a review. Now, in terms of the organization, the way the organs are organized, for the most part, the focus was on skeletal muscle in this chapter, so you must know all of this for the skeletal muscle. Okay. We didn't go into cardiac too much. Cardiac is very similar in its organization to skeletal muscle. Okay. There are some slight differences. Smooth muscle is quite different. We do not see that same kind of organization. We don't see the bundling. We don't see the fascicles. Okay. In terms of how these are going to be innervated, right, skeletal muscle, the neuromuscular junction, you really need to understand that. And in terms of the smooth muscle, then you just need to really talk about those varicosities. Okay. How these are controlled is important in terms of, are these voluntary or involuntary? And then what is going to regulate that control? Right? So even though the cardiac muscle is involuntary, right? We don't control how fast, we don't control our heartbeat, right? But our nervous system can have an effect on how fast or how slow our heart beats. Okay? Same thing with the smooth muscle, right? You cannot consciously control your smooth muscle in your intestines, but depending on what's going on in your body, it will speed up or slow down. Right? If you eat a big meal, it's going to be stimulated more to move things through. Right? If you're starving, it slows down. Now, in terms of the skeletal, you must go through and you must really be comfortable with how calcium regulates it. Right? The similarity between skeletal and cardiac here is pretty much the same. Right? We have not gone into that portion for smooth muscle, and we will not. So just a good way to make sure that you are getting all of the information.